I'm Alice Popejoy, an assistant professor at UC Davis in the Division of Epidemiology in the Department of Public Health Sciences. And today I'm going to be talking to you about population-based metrics for diversity and how that shapes human genomics research and reference resources. I'm my presentation here in Davis, and I live in Sacramento, and I do work in both places. And we're sitting on unceded lands stolen from Native peoples for the University of California campuses. So I want to acknowledge and honor the original inhabitants of the regions who came before the colonizers, whose harmful actions I have benefited from, and offer gratitude to their descendants for upholding their ancestors' traditions and stewardship. I also hope that when we discuss these topics today in this week of LabRoots Genetics Week event, and in general in our workplaces and in our communities, that we have courage to sit with discomfort and examine our assumptions, that we have gratitude and humility and openness to other ways of knowing, as well as committing to being trustworthy partners for the communities that we seek to engage in our research and our work. What does trustworthy mean? I'll talk about that a little bit throughout today's presentation. And to me, in the context of biomedical research and genomics, trustworthy means that what we're doing is accurate and precise, that it's beneficial and responsible for the impact of what we're doing, and that the research itself is just, diverse, and inclusive, as well as the teams doing the work. So in general, ethical frameworks and bioethical research have been slowly making their way into integration into biomedical research and are integrated at various capacities in different projects. 2 years ago at NIH, the Office of Science Policy held a workshop on integrating bioethics into biomedical research. And it was really important that they came out with this statement that said that really the goal of bioethics engagement is to make science better, increase inclusion of participants, stakeholders, societal values, and also to engender greater trust in biomedical research as an enterprise. So we're all about building relationships and structures that can enable bioethicists and biomedical researchers to collaborate together to advance our science. And this is some of the work that we're doing in the Human Pangenome Reference Consortium, or the US Pangenome Project. We have an embedded ELSI framework, or ethical, legal, social implications. So the embedded ELSI team is involved in all the different working groups that are producing a pangenome reference, or an updated human genome reference, from collecting data to the sequencing and production assemblies, and also especially how we do outreach to different communities of stakeholders, whether they're clinicians, researchers, members of the public, and especially, of course, users of our tools and resources from the consortium. So the embedded ELSI team is a team of many different scholars from different disciplinary backgrounds and perspectives. And we've been thinking about this question of how we can represent global genomic diversity in a graph-based data structure, potentially, as the genome is going to be in this graph-based pangenome structure. How are we going to sort of match that with how we represent diversity? And maybe we just look at genetic similarity, but what we're currently doing in research and medicine as far as population descriptors and how we use them to measure diversity is not working. So I'll talk about that in a moment. Some of the elements of what we're doing in the HPRC, or the Human Pangenome Reference Consortium, have to do with lots of different issues, from the technical and logistical to the ethical, legal, and social implications, as well as the regulatory and legal and financial and commercial elements. So one of the questions that we think about, and what I'm focusing on today, is how we define population diversity. And then from there, for the project, how do we select and prioritize individuals for sequencing and inclusion in a new pangenome reference? How do we represent those populations actually in the reference resources and the data sets we make available, or do we want to? So there are lots of different questions. You can read them here on the slides, and they will be available later. And of course, if you have any thoughts about this and want to collaborate and work with us, this is an open invitation throughout. So keep that in mind as we go through. So I mentioned first that if we want to be trustworthy as researchers in building our research programs and our reference data sets, we have to be accurate and precise. So we'll talk about defining populations and then looking at downstream impacts of when we stratify people. We also want to be beneficial and responsive to the communities that we serve and in which we work. So we want to partner with people to include as many different perspectives as we can in the research, and also to maximize benefit and knowledge sharing in a bidirectional kind of way, not just the researchers and physicians know everything and they're communicating to the public, but that we're learning really valuable things from non-scientists as well. So we want to essentially promote justice through ethical decision making in science, but this also makes our science better, especially when we can use methods that are respectful of our participant and partners' perspectives, identities, and the expertise that they bring to the table about their own lives. For example, social determinants of health. We're going to learn the most about that from people themselves. So first, accuracy and precision. Well, I've given lots of these talks in different places over the years. This one is a result of a talk I gave to the National Academy's Committee on the Use of Population Descriptors in Genomics Research last summer, and the report is now out. I encourage everyone to take a look at that report and the decision tree and thinking about population descriptors for their research. But what I wanted to highlight here is that when I ask a group of clinical geneticists, researchers, et cetera, about what comes to mind when they think of a population, almost always a group of people comes up in the center. So no matter how we think about grouping people into populations, it's important to actually take a step back and realize that when we say the word population, we just mean a group of people, however that might be defined. So I've also asked these questions about what word or words come to mind when you think of ancestry, ethnicity, and race. And what I've learned from doing this is that most people think of ancestry in terms of family and genetics and lineages, maybe geographic origins, whereas ethnicity is thought of really as a way to group people by culture. And race is considered a way to group people by physical appearance and specifically skin color. We say social construct, but I think sometimes that sort of gets used to not have to think about what it actually means and how it is being implemented. It is indeed a social construct, and it's meaningful in that we use it in a lot of different ways, but maybe not in exactly the right ways. So here's just a summary of the different ways in which we might think about these terms that get thrown around and often conflated with one another in our research and clinical practice: population, ancestry, ethnicity, and race. So anyone who spent any amount of time in the US knows that we use these racial and ethnic classification frameworks from the US Office of Management and Budget. These are familiar terms probably to many people, although in places outside the US, they may not be as meaningful. For example, Hispanic or non-Hispanic makes sense in the US context or even in the UK, but in Latin America, it's not a term that's really used at all. And so I think it's really important to take a step back and consider the words that we use to group people and what do they really mean. Like Asian. Geographically speaking, the census population size of people in the countries that are considered Asian countries is more than half the world's population. So what information is that really giving us about somebody's origins, let's say? The Venn diagram on the right here just shows us that there's a lot of fluidity and overlapping concepts between these terms. So it's really important to understand their limitations. Also to understand that NIH is required to collect the demographic breakdown of who's included in federally funded research from investigators using these OMB categories. Now, luckily, the OMB and the Census Bureau are in the process of updating the categories, but it still may not be useful for people's research. So a sort of unfortunate consequence of the congressional law in 1993 requiring this reporting of diversity, which was actually had good intentions. The idea was to track who was included in biomedical research and make sure that we had representation from from folks other than white men, who were primarily those included at the time. But a sort of unintended consequence is that once researchers go to the trouble to try and figure out how people in their sample fit into these social categories used in the census, that's pretty convenient to use in a stratified analysis, you might say, for genomics. So I think it's really important to understand the history of why we do the things that we do and what we use, so that we can start to do something a little bit more precise and accurate moving forward. So what factors contribute to one being identified with any population, group, or category? Well, you have to think about how the category is defined, who defines it, what the purpose is of categorization, what the context is, who decides whether someone belongs in a category or not, and what are their motivations. And then, of course, how are the data actually ascertained? What's the framework for data collection? Is there an open-ended option? Is it just multiple choice? And I think an important question to ask is whether people really have autonomy and agency and feel safe to self-identify in the ways that they would want to. So let's take Hispanic, for example, looking at it across contexts. If you look at the US Census categories, this term did not even arise until 1980 on the census. And there are different elements of ways in which this is constructed. So there's time as an element of difference. There's also where you are in the world, and who you are and why being asked. So in Brazil, the categories are yellow, white, indigenous, brown, and black, which may seem kind of shocking to us in the United States, just as probably shocking or confusing our categories might be to an average Brazilian citizen. In Mexico, there's a unifying national identity. All the indigenous identities have been sort of stripped from their census, and there's this real effort in the last 100 years or so to unify the country with one identity. In Argentina, it's similar in that there's sort of a unified idea, but it's an idea of mixture, this crisol de razas, or the melting pot. And why someone might be categorized is different. Somebody who is identifies as Hispanic and also as white, or could be considered white passing, may choose to identify as Hispanic on a job application if they feel like it might benefit them, and then change their answer or have a different answer when going to the doctor or enrolling in a biomedical research study, for example. It's also time dependent of when this term arises. In the US OMB context, the category Hispanic, Latino, or Spanish origin includes all individuals who identify with one or more nationalities or ethnic groups operating in Mexico, Puerto Rico, Cuba, Central and South America, and other Spanish cultures. But where and when were the Spanish cultures? You cannot separate the idea of grouping people into categories, especially with terms like Hispanic that are relative to other places and other cultures over time, from the history of colonization by Europeans. So if you look at these different maps on the bottom right of which European countries had colonized Latin American countries, which gives it the name Latin American, it would be different based on what time frame you're looking at. So it's really important to understand, again, the history of what we're using and why, so that we can start to be critical in evaluating what we're doing today. And of course, there is a very long and painful history of racial classification in the US that's rooted in a notion of biological or scientific racism, which was initially constructed a very long time ago to justify the horrors of slavery and biocolonial extraction of resources, to oppression and even genocide. So I think it's very important that we understand that having conversations about this is going to be very uncomfortable, and also extremely, extremely important given the recency, especially in the US, of overt laws that segregated people on the basis of skin color. And this, of course, is rooted in an idea that there's a fundamental biological difference between people, even at the genetic level, on the basis of how they are identified externally by racial or ethnic categories. So in the Jim Crow era, we had explicit segregation of people who were identified as white and Black on the basis of skin color. And the justification for that was given was that there needed to be different medicine for different types of people. But this is the typological thinking that the National Academy's report, which I mentioned, is urging us against. And of course, we know from decades of careful human genetics research that there is no genetic basis for these racial categorizations. So Dick Lewontin, for example, in 1972, looked at the variation across within and between these sort of traditional racial categories. And he found that the vast majority of the variation was within each of these groups, not distinguishing between them. So I really like this Venn diagram from Alan Donovan's work in science education, and you see that most groups, as we call them, in these racial groups, but also at sort of even if you're grouping people by ancestry, continental level ancestry, which somewhat approximates those categories, all groups are genetically alike across the board because we all share the same sets of differences that are within those groups. So most of the human genome is identical for every single one of us. But in those tiny places where we differ in similar ways in different places, but maybe just in slightly different amounts. So let's ground this in an example. The Duffy blood group alleles, for example, do have some geographic differences in how they're distributed, but it's not as if there are some alleles that are only in one place or only on one continent and absent everywhere else. In fact, the Duffy blood group alleles are present across every continent, just at different relative frequencies. So you have these geographic clines of allele frequencies, but none are unique to any one continent. So it wouldn't be fair to say, even for the one that is selected for because of its beneficial effects in protecting against malaria, that this would be an African variant, because it is, of course, present in other parts of the world where malaria is endemic. So it's also important to understand that when we're in a clinical context, doctors are not able to look at something like genetic ancestry. That's not even a concept that is that's widely understood, I don't think, across a lot of, even in the genetics community, we think of genetic ancestry as something that's very concrete and specific, but really it's also a relative measure, because if you go back far enough, we all share genetic ancestors, and we all are part of the same human family. So genetic ancestry itself is a relative measure, and it's an estimate or a proxy for something like genomic background or maybe genomic similarity. And even that is not what's being used in clinical practice. So the Ancestry and Diversity Working Group of ClinGen put out a paper that we wrote on a survey, which included questions for clinical genetics professionals and researchers on how they might use population descriptors like race, ethnicity, or ancestry. And the vast majority told us that race or ethnicity is reported directly to them by the patient, or they retrieve this information from a patient's medical record. And 18% of the survey respondents told us that this information is reported by another care provider, possibly without even asking the patient directly. And only 4.6% told us that they actually infer genetic ancestry from a patient's DNA sample. And when you look at the forms that clinical geneticists use to send tests off to the labs, there is a compendium of categories that are used, some very few in certain labs, some have quite a number of categories, but the most granularity that you see is in Europe. So Northern European, Southern, Western European, there's all this specificity around Europeans, which actually are very genetically similar, roughly speaking, compared to lots of other parts of the world. So It's really important also that the categories that we use are intentional, that they're actually telling us something. For example, Ashkenazi Jewish or French Canadian makes sense because those are founder populations where there's a very small number of individuals who sort of founded the population. There was a reduction in size and then a later expansion of the population such that the amount of genetic diversity is pretty small, and there was a just random chance that some harmful mutations proliferated in those populations. So a clinical geneticist might really want to know if you have that ancestry. But something like Asian on one of these forms, again, half the world's population, maybe not so useful. Again, even when we're using genetic ancestry, as they do for the NOMAD database, which gives us information about allele frequencies in different populations, and how those populations are defined actually is using genetic ancestry and principal component analysis, which we'll talk about in a moment. And they report allele frequencies for these groupings. But again, how useful is that? If we only have, if we have a majority of our individuals in the data set are a non-Finnish European, which again has a really small amount of genetic diversity, we're missing a lot of the diversity that we really need across the globe to make accurate estimates, especially when we're dealing with point estimates like an allele frequency. Also, the way that the categories are constructed may not be that useful. You have African/African American. That could be a lot of different kinds of individuals, from people who are recent immigrants from African countries to the US who identify as African American, or people who are descended from individuals who were enslaved in the US, or people who are currently living in an African country on the continent. And there's so much diversity within the ancestral homeland of our species, which is the continent of Africa, that if you compare two individuals randomly from different parts of Africa, they're going to have more genetic differences on average than if you compared either one of them, let's say, to a European individual. So what does a point estimate like an allele frequency really mean for a category called African/African American? I don't know. Similarly, other reference data sets like the UK Biobank, which is used for a lot of genome-wide association studies these days, and they're expanding it, and they have phenotype data as well as genotype data. So it's a very rich data set in the sense of the type of information that they have. But who they have the information from, again, is very biased. Because there are orders of magnitude more individuals identified as white British than anybody else. And of course, these categories are kind of strange. They're what's used in the UK census. But in the US context, we wouldn't understand why Chinese wouldn't be considered Asian. So there's a lot of social, cultural, and political factors that go into how populations are defined. And it's just important to understand those caveats when we're using these pre-established reference databases. So there have been efforts to standardize population frameworks using things like principal component analysis. So the map on the right shows us that. But we're missing two key groups here that are just sort of called out in the paper as admixed groups, including different sort of Latin American cohorts and also the quote unquote admixed African American. But that term admixed sort of implies that only those groups are those who have mixed and mingled for since the beginning of the species, whereas these other regions are sort of intact ancestry populations. But that's not true. We've been moving around and mixing and mingling since the beginning of time for the species. So it's really difficult, and I've sort of abandoned any hope that we could standardize any sort of population framework, because the information that we need is different depending on the situation that we are invoking this idea of grouping people. So I wanted to address this principal component piece because it is very popular in the field of genomics to use as a way of sort of grouping or visualizing the amount of genetic diversity that you have in a sample. And a lot of this stems from this publication in 2008 where John Novembre and others were able to kind of reconstruct the geography of Europe using genotype data from individuals and coloring the individuals on the principal component map by the country where they were sampled from or where they were located. And this sort of works because there are these migration patterns into Europe in a south to north, east to west kind of way, such that the allele frequencies roughly correspond to latitude and longitude. So this looks really great. And a lot of people thought, wow, this is it. We can just use genotype data and principal component analysis, which is a way to reduce the dimensionality of a complex data set so you can visualize it in 2D. The problem is that this is a very particular case for Europe because of its migration history. And also, John Novembre followed up on that paper many times over, trying to implore people to understand that what people have sort of taken from that and run with it is perhaps not the best way to represent overall genetic diversity in human populations. And one of the reasons for that is he said that in order to even get this messy data cloud of people who all were very genetically similar into this geographic structure, they had to use over 100,000 loci. And even then, it only was a very, very small differences in those separations. So we're artificially sort of separating the differences to make them look bigger. So he says in a later paper that these methods like PCA or admixture analysis do a poor job of conveying absolute levels of differentiation, and that when we visually observe these genetic clusterings of people in this group, that we can be easily misled to believe that there's actually some deep differentiation between populations, even when the signal that we're seeing comes from these very subtle allele frequency differences at a very large number of genomic loci. So he talks about admixture, PCA, space mix, and then EMS specifically. But I think this goes for any sort of dimensionality reduction tool or tool to cluster people when there aren't necessarily natural non-overlapping groups. He shared that there can be misconceptions that arise, especially from how direct-to-consumer genetic ancestry tests are using continental groupings to describe people, and that we may mistake the outputs of these methods to believe that our variations in humans are really sharply divided geographically, and that there may be a common allele in one part of the world and not anywhere else, and that's just not typically the case. Finally, he says that we might mistakenly conclude that two people from different parts of the world mainly differ across their genomes due to alleles that are restricted to the places they come from. But that's not the case. People are going to just differ on average roughly at the same loci, and there are just these different overall average frequencies that do have these geographic clines. So we talked a lot about accuracy and precision, and we're towards the end, so I'll kind of race through the end here. But I think it's important that we think about how to be beneficial and responsive to communities that we're partnering with. So for example, what if we were to consider the pre-colonization California, which had over 300,000 people from over 200 tribes? What if we use these groupings to think about population diversity? We probably couldn't, because we don't have the data. A lot of this has been lost, and a lot of these divisions at the time were, again, very fluid as well. Or if we use Native Land Digital, which I encourage everyone to go to see where they are in the world and if there's information there about the native or indigenous languages or territories that they might be interacting with. What if we represented populations that way? I think there might be a very strong knee-jerk reaction, maybe even big question marks above your heads wondering why we would do this. But I think similarly to somebody who isn't already steeped in the culture of using race and ethnicity categories to represent ancestral diversity, the way we do things might also be similarly puzzling and unjustified. So it's important to understand, also why I bring in the need to pay attention to Indigenous communities, specifically in genomics. And it's because there's a long history, of course, of exploitation and of just severe atrocities against the American Indian Alaska Native communities by settlers and by the government. And in the genomics community, this has happened as well. There's a case of the Havasupai tribe in the Grand Canyon who actually won a lawsuit, or they settled a lawsuit with a university in the Southwest because their data had been used in ways that they hadn't consented to. So as a result, there's been a large reaction and a really fantastic response by the Indigenous genomics scholarly community to come up with a set of principles that could be used in addition to or alongside the FAIR principles for data use, like findable, accessible, interoperable, and reusable, to think more about how we can really respect Indigenous data sovereignty and integrate CARE principles. So this is the FAIR model where we're looking at policy, research, and infrastructure. But the CARE principles for Indigenous data governance are more about the people who are being impacted. So having collective benefit, having authority to control, having responsibility for all sorts of things, from the relationships to the worldviews of people that we're working with, and ethics to minimize harm and maximize benefits and to promote justice. So there are lots of different groups all over the world who are working on these types of frameworks, and I think it's really important that we consider them. So finally, how do we prioritize inclusion of diverse populations given all of these considerations of accuracy, precision, and respect for cultural values and worldviews? Well, one way we're thinking about it, or we have thought about it in different projects, including the Pangenome Project, is to look at global representation. Just how many people are in different parts of the world, and let's compare representation globally to who's represented in genome-wide association studies, as was done in this paper from 2019. So there's a good case to be made for this, that it's data-driven and the goal is equity and global representation. But a weakness is that we still fall back on these continental level categories, and it really all depends. These proportions depend on how you define them. So in the 1,000 Genomes Project, which we've primarily sampled from for the Human Pangenome Project thus far, shows different global variation across the globe. And again, in the gray slices of the pie charts, those are the common variants across all the continents. So that's important to keep in mind. And you see this elevated diversity in African genomes. So many of us have argued and I think this has been successful in the Pangenome Project to look at how much per individual variation is being contributed to the reference data set in order to sort of prioritize these for selection. And if you look more deeply into the 1,000 Genomes reference data set, you'll see that actually the way the populations are described are very specific. So African, Caribbean, and Barbados, African ancestry in the Southwest USA. And often these get sort of rolled up, so to speak, into the continental groupings, which you see in the pie chart, which come back to sort of equivalent to racial categories. And I would encourage us in the future to not strive for equality across these continental groups in the sense that you're treating them all equal, but to do this more equitably, where we would preferentially sample from populations that have been underrepresented and that just simply have more genomic variation to contribute. So again, when thinking about how we can represent global genomic diversity while maintaining ethical frameworks and data sovereignty, we might consider mapping diversity across multiple dimensions, which is something that we're doing in my lab. So I suggest that we go from a categorical framework to a more open-ended framework that takes into consideration multiple dimensions of diversity. So in summary, and to conclude, population descriptors may have some utility, but they need to be very clearly defined and justified, especially when used in genomics. And now we have a resource from the National Academies to be able to do this. That data on clinical labs and just in general in these frameworks are highly heterogeneous and nothing is harmonized into a single standard. Also, that allele frequencies, instead of being these point estimates for a population whose denominator really defines who's included in the population, defines the denominator, and thus how big or small those frequencies are, that maybe we could look at distributions of traits across whatever categories it is that we're looking at, and just understand that those are really dynamic and relative measures. Also, the new pangenome reference, which is graph-based, is going to really change the way we represent human genomic diversity. And I think the same can be done with demographic data, and thinking more carefully about how we can respect multiple identities, even within an individual. So thank you very much, and I'd like to acknowledge the different projects and grants and collaborators that have participated, and Abhishek in my lab, who's been working on some methods for open-ended identification, and the references are all here as well. So at some point you'll have access to these, and you can do more reading about this yourselves. So thanks so much.